All right, everybody. Thank you for joining us today. We're going to go ahead and get started. It is 12 o'clock on the dot. My name is Shannon Spear. I'm an assistant professor at the University of Arkansas, focusing on water quality research. And in partnership with Lindsay Pease at the University of Minnesota and then MP Hayes at LSU, have put together this Basin Breakthrough series featuring early career leaders in Mississippi River Basin water quality research. Today, we're going to focus on the Middle Basin. We've got some wonderful speakers lined up here. But before we jump in, I've just got a quick set of announcements for y'all. Hopefully, you're seeing my screen here. But we have a new opportunity coming up in the early winter. So, January 27th to 29th at the University of Arkansas, we are hosting the Early Career Confluence Workshop, which is going to be focused on bringing together early career researchers working in the Mississippi River Basin to share their research, contribute to research agenda development for the entire basin, and also participate in some professional development opportunities. The workshop does cover all registration fees, meals, etc. And we can cover hotel accommodations for 3 nights for the first 20 applicants. So if you are interested, I will drop the link to register with more information in our chat here for everybody. But registration is due November 5th. And then we also have one more seminar after today. So we had our Upper Basin one 2 weeks ago. Today we're focusing on the Middle Basin, and then we will be focusing on the Lower Basin in our final seminar on November. So with that, we'll jump into our speakers for the day. So we have three speakers, Olivia Bongiovi from University of Missouri, Anna Vincent from Northwestern, but presenting research from Indiana, and then Emily Byers from the University of Kentucky. The ground rules for today, each speaker will be given about 15 minutes to speak with about 30 minutes for questions. As we work through this, you all can enter your questions in the Q&A chat during the talks or right after the talks end, and we'll aim to answer as many live as possible, keeping in mind our approximate 18-minute time limit per person. And if we don't get to your question live, then the speakers will have the opportunity to respond in the chat as well. And so with that, I will go ahead and stop sharing my screen. And Anna Vincent will be speaking first today. So I will let her go ahead and share her screen while I introduce her. Um, so Anna is a biogeochemist and an ecosystem ecologist and is currently a postdoctoral scholar at the Northwestern Argonne Institute of Science and Engineering at Northwestern University. She completed her PhD relatively recently at the University of Notre Dame under the direction of Jennifer Tank, where she assessed the controls on inorganic nitrogen cycling and the role of restoration and conservation in mitigating changes in stream metabolism following storms in agricultural streams. Her current work is focused on using green infrastructure to monitor climate change dynamics in urban environments and using high frequency sensors to track pollution hotspots and hot moments in urban streams. Throughout her seminar today, Anna will present her work exploring linkages in ammonium and nitrate transport in working agricultural landscapes and her examination of the effect of cover crops as a mitigation strategy to prevent ammonium losses at the field and watershed scale. And with that, Anna, you can go ahead and take it away.
Great. Thanks, Shannon. And if you can just give me a head nod or a thumbs up that you all can see my full screen. Perfect. Okay. All right. So thanks everybody for being here today. And many thanks to the organizers of the webinar for their invitation to present. So as Shannon mentioned, I'll be talking about mismatches between ammonium and nitrate signatures at different spatial scales and looking at how different controls on nitrogen loss in different agricultural watersheds can lead to different patterns. So before we dive into any data, I did want to take a moment and thank all of my co-authors and collaborators at the University of Notre Dame, where I was when this work was completed, as well as all of our funding sources and collaborators, those farmers and landowners in our watersheds. And all of the data that I'm presenting today are available in a publication with Agriculture, Ecosystems, and the Environment. So to start us off, we know that synthetic nitrogen fertilizers are added to agricultural watersheds to help maximize crop yields. However, in the Midwest, anywhere from 15 to 65% of the supplied fertilizer, typically added as anhydrous ammonia, is lost to adjacent waterways. And this is ultimately done in combination with this fertilizer use. These watersheds are engineered specifically to help facilitate the removal of water from the landscape into adjacent waterways. And this helps to maintain high productivity and those high crop yields. So this is done through two primary modifications. The first is the installation of subsurface tile drains, the second being stream channelization. But ultimately, cover crops or other conservation practices can help mitigate nitrogen losses from these watersheds that contain these modifications. And so one such practice is planting winter cover crops. And these have been shown to assimilate inorganic nitrogen into biomass, which is what we're seeing here in this plot from Christopher et al., or even a marginal increase in cover crop biomass results in a drastic reduction in that soil nitrate pool. Cover crops have also been shown to help prevent erosion and improve soil health across working watersheds. And in terms of water quality, they've also been shown to help reduce tile drain nitrate and SRP losses, though watershed scale results do tend to be more variable. And while this is all fantastic and a great stride forward, the effect of cover crops on ammonium losses are not well documented, but this is a really critical linkage to understand, given the biogeochemical relationship between ammonium and nitrate via the process of nitrification. Now, to add to this complexity, the climate in the Midwestern US is changing with climate change. So storm intensity and frequency are both increasing in this region, and this makes it really important to document the timing and the drivers of ammonium export from agricultural watersheds to help us understand how inorganic nitrogen transport responds to the changing climate. So in this study, we asked the question, how does conservation influence ammonium export? And we divided our hypotheses according to scale. So thinking first about the field scale or soils, we predicted that cover crops would effectively assimilate ammonium into biomass or soil organic nitrogen. At the edge of field scale, meaning those tile drains, we predicted that the assimilation of ammonium on the fields in soils would reduce tile losses, and this would alter DIN stoichiometry, so that relationship between ammonium and nitrate coming out of tile drains. And then finally, at the watershed scale, we predicted that the magnitude of ammonium yields would be lower than nitrate due to preferential ammonium removal, but that the timing of ammonium and nitrate yields would both be linked to high flows, so storms being an important component of export. So this study was conducted as part of the broader Indiana Watershed Initiative, where we had two working watersheds in north central Indiana. First is the Shadow Ditch watershed, which I have noted on my slides moving forward as SDW in Kosciuszko County. That sits at about 1,300 hectares. And then a little further west, we have the Kirkpatrick Ditch watershed, or KDW, moving forward in Jasper County, and that's about twice the size at 2,600 hectares or so. Both watersheds are dominated by more than 85% row crop agriculture in a corn-soybean rotation, and they both contain those modified hydrology techniques. Those tile drains are installed, and there's quite a bit of ditch channelization. And then finally, we did have variable cover crop coverage throughout the study period. So that ranges from 2016 through 2023 at Shadow and 2022 at Kirk. So in terms of our field methods, we collected our soils and cover crop biomass twice a year, in fall after the cash crop harvest and in spring before the new cash crop was planted. We sampled both watersheds twice a month for water chemistry, and so that included grab samples from stream sites and tile drains for ammonium and nitrate analyses. We measured instantaneous tile flow, and we had USGS gauges installed at both outlets to measure daily flow. For our tile drains, we estimated an instantaneous nutrient loss. So that's taking our concentration, multiplying times our flow, and that gives us a nutrient loss as mass per time. And then at the watershed scale, we used those nutrient grab samples from our stream sites, mainly our outlets, and our daily flow data to model daily ammonium and nitrate export. Later in the slides, when I show those watershed scale results, those values will be expressed as a yield. So looking at it as kilograms per hectare per day to help us normalize between those two watershed areas. So diving into some data here, first looking at the field scale or those soil results, we see right off the bat that soil ammonium and nitrate content or those pools were occurring on comparable scales. So those y-axes there fall into a similar range. And then across the x-axis, that is the cash crop planted during the prior season. So it was the field a cornfield or a soy field. For our ammonium results, we see that cover crops did reduce soil DIN for ammonium by about 57% at shadow. We did see a negative trend at Kirk, though it wasn't significant. And when we bring in the results here for nitrate, we saw 59% reduction on cover crop fields for shadow, 65% reduction at Kirk. So this is suggesting that cover crops do effectively reduce soil DIN, and this is happening sort of across the board. So regardless of what that prior season's plant was. We were also able to confirm the negative trend between soil nitrate pools and cover crop biomass that was documented in the Christopher et al. Paper. But we did not see the same relationship between the soil ammonium pool and cover crop biomass for ammonium. So that's suggesting that there is some sort of different driver or mechanism behind that removal. And so at the field scale, cover crops effectively reduced soil ammonium, but variation in cover crop biomass was only explaining the soil nitrate pool. So next, moving on to our edge of field results. So thinking about those tile drains, this slide is going to have a lot of data. So I'm going to walk us through it. So first, we're going to take a look at just the tile ammonium loss results. This is as micrograms per second. Across the top of the slide here, we have our different study years ranging 2016 to 2023. And then the pink rows are the shadow results, purple are for Kirk. Again, we'll look at ammonium first, and then we'll bring in the nitrate results. So off the bat, we see that cover crops reduced tile ammonium losses by anywhere from 67 to 98%. But these patterns and years differed between the watersheds. So at Kirk, for example, for ammonium, we saw a 64% increase during one of the study years and no difference across the other years. Bringing in just the title for our next set of rows, that tile nitrate loss, notice the difference in the scaling here. So the nitrate results are in milligrams per second, where those annual tile nitrate losses are about 1,000 times higher than they are for ammonium. So we see a difference in scaling emerging. We saw that cover crops reduced nitrate losses at shadow by anywhere from 45 to 94%. But again, that effect at Kirk was more variable, ranging from a 93% reduction in 2021 to a 70% increase in 2020. So generally, the reductions between the ammonium and the nitrate losses are occurring during similar years between or for an individual watershed. But when we pair or pool those years together to take account for some of that interannual variation, we see that there is a stronger signal of cover crops at shadow, where we saw that reduction in both ammonium loss and nitrate loss compared to Kirk, where we were only seeing that signature for nitrate loss, but not for ammonium. So for our tile drains, we saw that cover crops can reduce ammonium losses, much like they do for nitrate, but that effect is susceptible to interannual variation. And then third, moving on to our watershed scale results. First, I just wanted to show you what runoff looks like in our two watersheds. So this was quite variable across our study years, but it was comparable between the two watersheds. Note that those panels are showing runoff in a declining pattern across the years, but those years aren't necessarily in chronological order. So I just wanted to point that out. And now let's take a look at the nutrient data. So what did ammonium yield in the top row of this figure, and then cumulative nitrate yield on the bottom? What did that look like? Much like with our tile results, we did see a difference in the scaling at the watershed scale. So the magnitude was very different. And we saw that ammonium yield was higher at shadow for most of our study years compared to Kirk. And the opposite pattern emerges for that cumulative nitrate yield, where it's higher for most years at Kirk relative to shadow. So this is suggesting that environmental context matters. These two watersheds, I have a picture of each of them in that bottom right corner. They look very similar, but we know that there are differences in their soil composition. For example, clay does tend to be more prevalent at Kirkpatrick. We had higher ammonium in the groundwater at Shadow, which could be helping to explain some of the patterns we're seeing here. And we also did have a difference in discharge, where discharge did tend to be higher at Kirk relative to Shadow. These orange boxes now are highlighting the difference between the fallow season when those fields are bare relative to the growing season. And we see that most of that export of both ammonium and nitrate is occurring during that first part of the year, or that fallow season starting on October 1st. We see more of a step function, or like a pulse pattern emerging for our ammonium yields relative to a more constant accumulation for nitrate. And this becomes really apparent if we take away these plots looking at it over time, and instead we look at cumulative ammonium and nitrate yields relative to runoff. So nitrate is exhibiting that near linear relationship between cumulative nitrate yield and runoff, whereas we see that pairwise step function increase for ammonium. So while nitrate is leaking out steadily, it's sort of consistently exiting the watershed. Ammonium is actually pulsing out of the system, and it's stimulated by those strongest storm events. So at the watershed scale, both ammonium and nitrate export occurred at different magnitudes, much like they did at the tile drain scale, and ammonium export was closely linked to the strongest storm events. So what does this tell us? We know that cover crops can effectively mitigate ammonium losses, but watershed characteristics such as things like seasonality, hydrology, land management are really important when understanding and documenting these patterns. And it's suggesting that environmental variation can mask the signal of cover crops for ammonium. So at the field scale, we saw that cover crops reduced soil ammonium and nitrate, but different drivers were emerging. So while it might be related to biomass for nitrate, it could be more related to soil chemistry, things like clay content and such for ammonium. At our tile drain scale, cover crops reduced tile drain ammonium losses, but this was susceptible to soil chemistry, flow, environmental conditions present in the watersheds. But we did see sort of a difference in scale emerge between ammonium and nitrate loss when we started to look at the water chemistry. And then at the watershed scale, we saw that storms emerged as an important driver of watershed scale yields, but watershed scale export was still dominated by nitrate, which has been shown previously. So just as a final point, sort of a plug for positivity, we did want to note that soil nitrification seems to have a potential impact on the scale of our ammonium and nitrate losses. So not only are using things like cover crops or planting cover crops during the fallow season, that's really critical to helping mitigate the effects of nutrient losses on water quality, but there's the potential for sort of a 1-2 combination to help limit nitrogen runoff and maintain productivity. So combining planting winter cover crops with something like a nitrogen stabilizer such as NSERV, and that can help mitigate those rates of soil nitrification, which would be the conversion of that ammonium into nitrate, which is then available to leak out of the system. And so with that, I'm happy to take any questions. As Shannon mentioned, I'm happy to respond to others in the chat later on if we run out of time. And with that, I'm happy to take questions.
Awesome. Thank you so much, Anna. That was a really great talk. Exciting to see. This is a photo that I took, I think.
It was.
Anna and I were PhD students together, at least way back when in my life. But well, since no one has put any questions in the chat yet, I do have a question for you. So you alluded to the role of nitrification at the end. Do you think that's potentially why at Kirk specifically, we see that relatively low ammonium and then much higher nitrate? Could that be the role of nitrification? And have you collected any data to maybe confirm that?
So yeah, that's a great question, Shannon. Thanks so much. That definitely is part of it for sure. We do know that in both of these watersheds, there is pretty high nitrification happening. I don't have any data for nitrification in soil. We did measure it in the streams, the stream sediments, and we did have relatively high nitrification rates for both Shadow and Kirk. So that I think is part of the story. I think the other major player here, particularly with those cumulative yields, has to do with the soil chemistry as well. Not soil chemistry. The groundwater signal. I think that much higher ammonium yield happening for most of those years at Shadow is the infiltration of high ammonium groundwater coming in from the hyporheic zone relative to Kirkpatrick. So I think that also is playing a role.
Awesome. Excellent. Well, I don't see any other questions in the chat, but if you do think of one, please feel free to enter it in, and Anna can go ahead and answer them as they do come in. I know sometimes, oh, look, we have one already. We'll ask this one, and then any more we'll let be answered via typing in the chat afterwards. But Jason has asked, could you re-explain the two graphs for the pulse trends observed in both watersheds? I'm guessing this refers to the cumulative plots with runoff.
Yeah. Yeah, absolutely. So uh, here on this slide, this is showing the cumulative ammonium yield on Y or cumulative nitrate here for the bottom two panels, and then comparing it to annual runoff for that year. And so what we're seeing is that with with ammonium, you see some of these sort of step function increases where ammonium is relatively consistent, and then as runoff is increasing, you end up with this jolt upward. And it doesn't happen for all years or all the time, but you do see these instances of these strong increases in ammonium yield happening very quickly as runoff goes up. Compared to this more consistent linear pattern that we see for nitrate, this is why we're thinking that it potentially has to do with individual storm events that are stimulating sort of the movement of ammonium from soils into the streams, which is where these data are coming from. Now, I did want to bring in the other panel. So this shows it according to runoff. But if I go back to the last slide here and I can pull off, there we go. Here, what we see is a similar thing where you have these step function increases, and this is with time. So these increases where it's happening very, very quickly, these are individual storm events where we're seeing a large jump in the cumulative ammonium or nitrate yield happening in a very short amount of time. And so this, combined with the relationship not being totally linear with runoff, is suggesting that it's more of a pulse addition of ammonium happening when storms come through that are really strong, that dump a bunch of precipitation into the system, or things like snowmelt, where you end up with this huge release of water that goes into the streams, relative to what we're seeing with nitrate, where it's more of a consistent leaking through time.
Awesome. Thank you, Anna. If you want to go ahead and stop your screen share, we got one more question for you in the chat. If you can go ahead and just type in an answer there, that would be wonderful. We have next Emily Byers. Dr. Byers is a Kansas native, and she obtained her BS and MS from Kansas State University and her PhD from the University of Kentucky in Biosystems and Agricultural Engineering. And right now, she is a postdoctoral scholar with the Kentucky Water Institute at the University of Kentucky. Dr. Byers' research is centered around watershed scale processes governing surface water quality and the interactions of nutrients and emerging contaminants during water treatment. Today, she will be speaking about contaminant cocktail mixtures, impacts on nutrient cycling, and the need for adaptive and targeted water quality treatment. And with that, I will let you take that away, Emily.
All right, thank you for that introduction. Hopefully you're seeing my slides appropriately. But yeah, thank you for inviting me to speak today. As mentioned, my name is Emily Byers, and today I'll be speaking over work I did during my dissertation that was related to optimizing constructed wetland performance based on the contaminant cocktails that we're seeing in surface waters. And so the work I did was related to wetland treatment systems, which have been used very extensively as nature-based treatment methods to mitigate non-point source pollution, but particularly when we're thinking about nitrogen and phosphorus treatment. Additionally, there has been lots of work done in this space looking at different ways to optimize treatment performance based on, wetland design, either to, optimize that treatment performance based on water quality conditions, but also taking into consideration the available land space we're working in or the climate conditions you're within. So, for example, we could think about a traditional land-based free water surface wetland where we have those plants actually rooted into the soil and in zones of standing water. And say we could compare that to a more emerging type of technology, a floating treatment wetland, where you have this vegetated floating mat that floats on top of a water surface, such as a pond or a lake or even a river. And then you have these roots freely floating within that water column, providing additional treatment. And so while these systems have been utilized to treat nutrients like nitrogen and phosphorus, obviously, we know these are not the only contaminants showing up in our surface waters and potentially then entering our wetland treatment system. And so if our focus is on treating ag water quality, there may be other things we're applying to the field, such as pesticides, or if we're applying manures, maybe there's antibiotics that we gave to the livestock, or even if we're applying biosolids, there could be human use pharmaceuticals. And all of those could be ending up in our surface waters. And so it's important to understand how these nitrogen mitigation strategies may be impacted by these other contaminants that are present. And so the work that I did was really interested in identifying common surface water contaminants and also in identifying their sources of contamination based upon the predominant land covers and land use practices present within Kentucky where this work took place. And so to do this, I analyzed surface water conditions across 4 watersheds. One that we, identified as predominantly urban in the central portion of the state. We had a more ag watershed on the western side of the state, and then 2 forested watersheds in Appalachia. However, one had higher activity of mining, and the other had a higher density of oil and gas well drilling. And so monthly sampling of these streams took place during the growing season from March to October to see if we could quantify the major contaminants that were showing up during this time period. And so I just want to really briefly highlight some of these results here. So focusing in on the pesticides that we analyzed, we identified 3 main pesticides. However, they each showed up in different watersheds. And so imidacloprid, for example, we found both in our ag systems, shown here in these red bars, but also in our urban watershed. And more importantly, the more toxic byproduct of imidacloprid was at higher concentrations in our urban watershed. And it's commonly why this pesticide is often considered an urban use signature pesticide. And then, of course, not surprisingly, in our ag system, we saw the highest amounts of atrazine and allichlor kind of peaking after that spring flush event, where this blue line is showing total monthly precipitation. However, something a little bit more unexpected happened in our forested, we'll say mining watershed, and that's when it had the highest amounts of glyphosate that we observed. And it really peaked in late July after a 1 in 1,000 year flooding event that devastated the region. And so it's estimated that approximately 9,000 homes were damaged, where entire homes, sheds, garages were washed away into the stream. And so presumably you could think that any sort of contaminants that were stored within those buildings was also spilled into the stream and could have contributed to environmental contamination. And so that could be why we're seeing, such greater concentrations of glyphosate peaking in this region after that flooding event had occurred. And then the last class of contaminants I'll briefly highlight are personal care products and human use pharmaceuticals. And so as an example, this graph here is showing caffeine concentrations in nanograms per liter. We're in our rural forested watershed that oil and gas, we'll say, had the highest amounts of caffeine. Well, actually, our agricultural watershed had the second highest amounts. And in both of these watersheds, they also showed higher amounts of other pharmaceuticals like acetaminophen, so Tylenol, as well as cotinine and human use antibiotics. And so this was kind of surprising to us because both of these watersheds are predominantly rural. So only about 5% of the watershed could be, defined as residential area. However, we're thinking that we're seeing higher concentrations in these rural regions compared to our urban watershed because of older wastewater infrastructure or non-centralized systems like septic, or what's shown here is a straight pipe, which is a common practice in Appalachia, could be discharging improperly treated or untreated waste into the stream and contributing to these higher amounts of pharmaceuticals in these rural regions compared to our urban watersheds where predominantly they use, traditional wastewater treatment technologies. And so while I mentioned, these wetlands are really good at mitigating nutrients like nitrogen and phosphorus, there also has been work done in seeing how these wetlands could work to mitigate some of those more emerging contaminants like pesticides and pharmaceuticals I just discussed. However, most of this work was done looking at those contaminants individually, and there's really a lack of understanding of how these contaminant cocktail mixtures may be impacting overall wetland performance. So specifically, how may they be impacting nitrate removal processes? So are we optimizing complete removal of nitrogen from our system through denitrification? Or is our inflow water quality parameters and wetland design facilitating more temporary removal of nitrogen, either say through plant uptake or microbial assimilation and soil absorption? And so depending on what transformation processes or pathways are predominant, it's going to change how we need to manage and maintain these systems to ensure their long-term success. And so because of this, the goal of the study was really to improve our understanding of the cumulative use of these increased commonly detected contaminants on nitrogen transformation processes within wetlands. And so to do this, I conducted mesocosm studies within a greenhouse setting, looking at those two main types of treatment wetlands I discussed earlier. So those floating treatment wetlands where you have this vegetated buoyant mat on top of the water surface, and then you have these roots freely floating within that water column. And I compared that to, say, the traditional freewater surface wetland, where you have those roots actually rooted into the soil and then standing water on top. And I compared both of those to soil controls to see if I could isolate differences that vegetation versus soil processes had on nitrate removal. And so for each of these mesocosms, I ran 6 different trials, where this first trial really just looked at nitrate removal to get a baseline understanding of how these systems were performing. And then I started adding individual contaminants. So say for our rural systems, I was looking at atrazine, glyphosate, and sulfate, which I didn't discuss, but we saw it at higher amounts in our rural mining watershed. And then for our urban systems, maybe we're more concerned with imidacloprid, caffeine, and PFAS. And so all of the concentrations that I added into these wetlands were what we actually observed in surface waters across Kentucky. And so I started out by adding these individual contaminants to identify which contaminants, or if any, were impacting nitrate removal. And then in the last trial, I added those contaminants together to see if it was really these contaminant cocktail mixtures and interactions that were impacting wetland performance, which is actually, what would be more realistic of the natural environment. So each of these trials took place over 10 days, where I took daily water sampling, and then also took plant and soil samples at the beginning and end of each trial to highlight if I could find out where those contaminants were moving throughout the different wetland zones. And so while those mesocosm studies can really show us the rate of nitrate reduction that's being impacted by these environmental contaminants and what proportion of those contaminants are moving throughout the soil, the plants, we really had this lack of understanding of what specific removal mechanisms were being impacted. And so because of that, I ran an additional trial where I also added the 15N isotope so I could trace nitrogen transformation processes throughout the wetland. So I did that by quantifying changes between the soil pool, or was the nitrate staying within the water column but just transforming into different species? Was it being taken up by the biomass and being stored within the leaves and the shoots, or was it being stored within those roots? Or were we getting complete denitrification or incomplete denitrification, and we're seeing this gas exchange of those nitrogen gases out of the water column? And so this method gives you a pretty reliable estimate of denitrification rates and also a strong understanding of these nitrogen transformation processes that may be impacted by these environmental conditions. And so getting into some of these results, first starting off with those kind of more emerging contaminants, we found that for caffeine, it was pretty efficiently removed from our wetlands, reaching 70% removal in just 10 days for our floating treatment wetlands, shown here in those solid bars, and then about 63% removal within our free-water surface wetlands. We also were able to detect caffeine that was taken up into the biomass and persisted until the end of the experiment. However, it only accounted for about 1% of the total enriched load we added, showing that caffeine was biodegrading and completely being removed from the wetland. And then as for PFAS, removal rates ranged from about 28 to 89%, where higher removal occurred within those floating treatment wetlands. And then of the PFAS that was removed, about 40 to 60% was being transferred into that soil and persisted until the end of the experiment. And so we're assuming that the rest of it was being taken up and stored within the biomass, and that's because we measured different analytes of PFAS within the water column and soil and didn't find any major degradation that was occurring, telling us that PFAS was staying likely in the form that we added it in, and was just moving through those different zones of the wetland. And then for imidacloprid, we actually added it at the concentrations that we weren't able to detect within the water column. And that was done to maintain those concentrations we saw within surface waters to keep this as realistic as possible. However, we were able to account for about 34 to 25% of the imidacloprid that was either taken up by the biomass or stored within the soil and persisted until the end of the experiment. And so of that, about 27% was taken up into the floating treatment wetland biomass, where more of it was occurring in the roots. And then for the free-water surface wetlands, about 19% of the imidacloprid was stored within the biomass, and more of it was occurring in the shoots and leaves. And then the remaining 6 to 7% was within the soil in both systems. And then for glyphosate and atrazine, we found that these were both rapidly removed from our wetland, reaching over 90% removal within just 24 hours of being enriched. And then as for plant uptake, there was a minimal detection compared to their enriched loads. However, what we did find is more of it was being stored in those floating treatment wetlands compared to those free water surface wetlands. And we didn't find any of these contaminants or their degradation byproducts being transformed into the soil and persisting until the end of the experiment. However, despite wetland design or contaminant present, these wetlands were still effectively able to remove nitrate from the wetland within just 10 days, reaching over 97 to 99% removal rates. And for a majority of the mesocosms, they saw significant removal by day 5. But when we started to compare the rate of nitrate removal, we do start to see some differences between the wetland design and the inflow water quality parameters. And so for the floating treatment wetlands, again shown here in those solid bars, they seemed to outperform the free water surface wetlands later on in that growing season, say after July, starting in trials 3 and onward, once these plants had matured and established. And so you can see differences in what the vegetation looked like from the beginning of the growing season to the end of the experiment. However, for the free water surface wetlands, they seemed to outperform the floating treatment wetlands earlier on in that growing stage, say during trials 1 and 2, showing us that there are significant seasonality effects on the performance of these wetlands. And so that's important to consider the timing of contaminants that would be entering the system. And so if contaminants are showing up earlier in the growing season, maybe we want to stick with these traditional free water surface wetlands, but otherwise floating treatment wetlands may be a viable option. And then when analyzing how removal pathways were impacted, we found that about 16 to 40% of that 15N was being taken up into that plant biomass, where more plant uptake was occurring within these floating treatment wetlands, and of that, most of it was being stored within those roots. And then as for gentrification, we were able to account for about 1 to 17% of that 15N was denitrified, with water playing the major part in that gas exchange. And then the rest of these gray bars are assumed to also have been denitrified 15N, but these plants were playing a much larger role in the diffusion of gas and getting nitrate out of the system. And then we also found that in the presence of these contaminant mixtures, there was decreased nitrogen removal rates, particularly for those free water surface wetlands, where those floating treatment wetlands didn't really seem to be as impacted when looking at overall nitrate removal rates. However, in the presence of both of these contaminant mixtures, we did see a larger amount of plant uptake, so that temporary storage of nitrogen compared to our controls. So in other words, while these floating treatment wetlands seem to have a high rate of nitrate removal, they stored a higher amount of that nitrate within their biomass, where the free water surface wetlands did seem to be slowed down a little bit in the presence of these contaminant mixtures. They still had higher rates of complete removal of nitrogen out of the system. And so while both of these treatment wetlands are viable options, since they had high nitrate removal and high contaminant removal as well, this feature kind of underscores differences in management plans that would be needed, particularly since those floating treatment wetlands accumulated more nitrate and more contaminants within their biomass. And so while our focus may be on, implementing these systems as best management practices to manage ag nutrient runoff, it's still important to understand how these performances are impacted by these other contaminants that could also be entering the system to ensure that we're designing them appropriately to manage the long-term success into the future. And so with that, I'd like to say thank you, acknowledge, our funding sources and all of our co-authors, particularly my advisor, Dr. Tiffany Messer, and I would take any questions you have.
Wonderful. Thank you so much, Emily. That was a really interesting talk. We'll answer one question for you that's in the chat live, and then we'll have you do any others via I keep calling it the typing option, but by typing the answer. The first question for you is from Kira. She asked, why is imidacloprid as nitro considered an urban signature while concentrations of imidacloprid tend to be higher in agricultural landscapes? Is there a mechanism or in urban systems driving its breakdown?
Yeah, so why imidacloprid is often considered an urban signature pesticide isn't necessarily based on my results specifically. It's commonly detected that imidacloprid is found in these urban settings, and that's because it not only is used to manage, insects, but it's commonly used in flea and tick medicine as well. So you have higher rates of domesticated animals also in these urban settings. And so it's important to understand, not just look at imidacloprid as a whole, but also these toxic byproducts. So are we seeing, longer pathways of imidacloprid, reaching the stream and having more time for it to be broken down rather than these ag systems?
Awesome. Thank you so much. You got at least one more in the chat. And another one just popped in. So I'll answer those. And you want to stop your screen share, we'll transition to Olivia's talk. All right, so our final speaker for today is Olivia Bongiovi, who is a fourth-year PhD student in water resources at the University of Missouri. She's currently working in the Hinkson Creek watershed, which is a mixed forested ag and urban basin, where she's working on parallel threads, focusing on long-term baseflow nutrient concentrations and greenhouse gas dynamics, specifically carbon dioxide and methane across the surface to land interface. On the method side, Olivia has adapted instrumentation for freshwater applications using a LICOR and smart chamber setup. With a collaborative group, she's also working on building a low-cost deployment chamber for intermittent streams as well. Olivia's goal is to generate measurements and insights that are directly useful for water quality management across the Mississippi River Basin. And today, Olivia will be talking about research updates from Mid-Missouri, nutrient chemistry, and stream greenhouse gas measurements. So with that, you can take it away, Olivia.
Hello, and thank you so much for inviting me. So I will preface, I am still a student, so this is a bit more in progress than the last couple of talks, but so just a little bit more about my background. I got my Bachelor's of Science in Biology from Bucknell University in 2022. So there's where I started my stream work, and I was working in agricultural streams. And I assessed the success of previous restoration in these streams. And then with another student, designed a project to test the efficacy of organic matter supplements in the form of leaf litter to complement riparian management in agricultural streams. The goal was to see if we could increase phosphorus uptake, and we did not, but it was an amazing learning experience. And I came to the University of Missouri in 2022, and I work with Alba Argerique. And right now, the working title of my dissertation is How does the expansion and contraction of the stream network affect carbon and nitrogen biogeochemistry? So the first half is about analyzing long-term trends in water quality in the watershed here. And then the second half is about understanding deal cycles of greenhouse gas in streams and potential driving mechanisms. So just to orient you to the system for those who may not be as familiar with Missouri. So Columbia, where the university is right in the middle of Missouri, and the Hinkson Creek watershed is the watershed that encapsulates Columbia, the city down in the south. So just some quick facts about the watershed. The stream is 26 miles long, and the total watershed area is 233 kilometers squared. So I would say a moderate size. It's a mixed land use watershed with agricultural land use up top, and it transitions to urban land use down in the south. It was listed as impaired by the EPA in 1998 for failing to meet water quality standards, and this was cited as due to general urban conditions and pollutants that do not support the expected level of aquatic life, and it contains high levels of E. coli. So this is what the land use map of the watershed looks like. If you kind of break it down into more just like agricultural, forested, and urban categories, it's about a perfect 1/3, 1/3, 1/3 split. And here you can see in that yellow and brown and green up top is where we have that more agricultural with forested pockets, and then all that red and pink down south. And there is a USGS gauge kind of down more in the urban center, and that is where all of my stream flow data comes from. And just because we have just that one site, I scaled the stream flow to the watershed area at each individual sampling point. So just to show that Hinkson Creek is extremely flashy, like a lot of urban systems. This storm is a couple years old, but I thought was a good one just to illustrate that. It went from about 15 liters a second, and then within a few hours was up to almost 34,000 liters per second. So the long-term project that we've been doing since 2018 in the Aquatic Ecology Lab at the University of Missouri is the synoptic sampling project. So we've created snapshots of water quality approximately every 3 months for the last 7 years. So samples are collected within a 2-hour window, and these are during, to say, we don't like to use base flow because it's hard to, what is base flow in an urban systems? We like to say stable flow. So we're looking for about stable flow for at least 2 weeks and no recent precipitation. And we analyze for several different nutrients and chemical parameters: dissolved nitrogen, nitrate, ammonia, organic carbon, total and dissolved phosphorus, soluble reactive phosphorus, chloride, conductivity, and the estimation of organic nitrogen. In my work, I focus on that organic nitrogen, that sort of black box of stream ecology, that those nitrogen-containing carbon compounds, the inorganic nitrogen, which we think of as more of the anthropogenic inputs, the fertilizer, the wastewater, and the organic carbon. So while I showed that graph of how flashy the Hinkson can be, again, the synoptic sampling does not target those conditions. It targets base flow conditions. So this data is about, my analysis is about a year behind because I've had to put this down to work on my greenhouse gas work. But this is what I have analyzed thus far. And those blue triangles are the sampling dates within the long-term stream flow. So we're kind of trying to target that middle 50%, certainly not the highs. So this is just a table of, we're seeing the average concentrations over the last, that's about 6 years of data. So it's not, the averages are not exceptionally high, but we've certainly seen some really high concentrations, especially we can see differences between the ag forested sites up north and the urban sites down south. And the star indicates that there is a significant difference in the concentrations between the ag forest and urban sites using a two-sample t-test. I like to point out that the highest DOC that we've seen is 41 milligrams per liter. That's super high. That was up north. And for inorganic nitrogen, again, it's not, the average isn't that high, but then the highest we've seen is almost 10 milligrams per liter. So these are graphs showing the average concentration of DOC, DON, and DIN between 2018 and 2023. From north to south, these are all of our sampling sites. And in that yellow, green, we see those highest concentrations. And we see a pattern in all three of them, generally, that it decreases as we move downstream to those lower purple concentrations. And then the overall calculated rate of decrease upstream to downstream is 41.7 micrograms per liter for carbon, negative 6.2 micrograms per liter of the organic nitrogen, and then a really small 0.5 micrograms per liter of the inorganic nitrogen. So we can see, but when we split this up by land use, we see differences. So in the ag forest reaches, the declines are much steeper, especially DOC, where we see the median is negative 113.6 micrograms per liter or 11.3 milligrams per liter. And the variability is higher, which is consistent with pulsed inputs like runoff, fertilizer, leaf litter, and lots of in-stream processing. So that microbial uptake and respiration, sorption, and more transformations. And then this is really what is driving this decrease upstream to downstream, because in the urban reaches, the median slope is zero for all three or near zero. So the concentrations are steadier and show little downstream attenuation. So mechanistically, this points to hydraulic alteration, like that channelization hardened banks that's typical of urban systems that reduces residence time in contact with bioactive sediments. And this can also point to continuous inputs like stormwater leakage, wastewater signals that flatten the profile and weaken the hyperaric exchange. So streams transport more than they transform. So the take home is that the concentrations decline everywhere upstream to downstream, but land use kind of sets the slope, with ag forest being a stronger transformation, and then urban is more of a steady conveyor. So when we're looking at the nitrogen fraction, which is a lot of what we care about here in the Midwest, we see that nitrogen concentrations are dominated by organic nitrogen in general. But when we're sorting this by stream flow, the organic nitrogen in orange, it's more of a steady concentration in the watershed, no matter what the stream flow is. And then we see that pulse of inorganic nitrogen with the highest concentration by far being at where we've sampled during the high stream flows, and the lowest concentration at the lowest stream flows. So then transitioning, then I was looking at, well, what is the nutrient concentration versus this space flow? So I use the SP model package in R to make a spatial linear model. And here on the bottom, I just have the organic nitrogen model. So this estimates the average effect of stream flow on the concentration on the DIN, DON, DOC across all sites. So the random effect allows that relationship, the slope of stream flow, to vary by site due to local conditions, land use, or geology. So this creates a random slope model. It acknowledges that while there's an overall trend, the strength or direction of that relationship might differ from one location to another. So starting really big picture, stream flow. So these graphs also will say, these are the site numbers 1, 5, 9, 13. So that top row is that ag forest land use, and these are just randomly picked from upstream to downstream. And then that second row, the 17, 22, 29, 36, that is in our urban land use, again, just randomly distributed across. This is the main stem here. So streamflow explains most of the variance in organic nitrogen, about a third, a little less so for DOC at about 0.2, and then lowest for DON at 0.1. So flow is a moderate driver overall, but certainly not equally for all constituents. And then by land use, this is where the pattern diverges. In the ag forest reaches, DIN rises strongly with stream flow, which is consistent with runoff and leaching, mobilizing inorganic, and during wet periods. In the same ag forest sites, DON and DOC are comparatively flat with flow, suggesting steadier sources and stronger in-stream processing, or a balance between dilution and mobilization. And then in the urban reaches, it kind of flips. DIN shows a reduced non-significant slope with flow, likely reflecting that flashy hydrology, short residence times, and possibly greater denitrification efficacy or lower fertilizer inputs. Meanwhile, DON and DOC increase sharply with stream flow in urban areas, pointing to event-driven pulses from stormwater outfalls, wastewater signals, and quick mobilization of organic material. Urban streams also start from a lower baseline concentration of DON and DOC, consistent with fewer natural organic matter sources. And again, those hardened channels could spike during stream flow. So kind of the big picture takeaway is inorganic nitrogen is flow responsive in ag forest systems, tracking hydrologic mobilization of inorganic N, whereas in urban systems, the big action is event-driven DON and DOC transport, which dominates transformation when residence time and hyperaric exchange are constrained. So, kind of moving more into the greenhouse gas deal cycle study. So, my big question was, what is the deal fluctuations of methane and CO2 in an urban stream system, and with the goal to capture fine scale temporal and spatial variability, and what are the mechanisms that drive these fluxes? So, we're focusing on FB, that is flat branch. It is the most urbanized subwatershed in the Hinkson Creek. It's about 90% developed. And this was of course, it didn't look this pretty in reality just due to physical constraints, but this was the map of our setup. So we had the Lycor soil collars. We had three transects, one where we floated it on the surface water, which I show over here on the right, and then one at the interface of the stream and the sediment there, and then back on the sediment away from the water. And we had a well at each one. And this is so we measured, it was more or less continuous. We measured every other hour between 4 a.m. and 9 p.m., and then once in the middle of the night between 12 and 1 a.m. So we measured those CO2 and CH4 concentrations. And then at the wells for the entire time period, we went a little bit more than 72 for some overlap, about 76. We took water samples from the wells and measured dissolved oxygen, both milligrams per liter and percent, the oxidation reduction potential, conductivity, temperature, and then the depth of the water to get at the extension of the water table. And then for a subset, so a little bit more than 24 hours, we collected and field filtered water samples from the wells and the surface water to be analyzed for total dissolved nitrogen, inorganic nitrogen, organic carbon, and soluble reactive phosphorus. And then on the upstream and downstream ends of our reach here, we had YSI exos collecting temperature, conductivity, dissolved oxygen, pH, and oxidation reduction potential. And then on top of the wells in the stream, we had Odyssey integrated light sensors for the estimation of stream flow. And I don't mean to leave you hanging, but we just were able to complete this the week before last. No exaggeration. Every time I've planned this for the last 5 months, it's the only time it rained in Missouri. In typical Midwest, just unpredictable weather. So I will say, if this is something that interests you, be on the lookout. Hopefully to present at AGU next year in a publication around the same time. And this was, I'll acknowledge, this is a huge effort. It took about 14 people working on this over those 3 days. So it's an awesome project. I'm really looking forward to diving into the data. And then as a collaboration across the Missouri Water Center, this is outside my dissertation, but the grant that I work on is Intermittent Streams Unveiling the Missing Piece of the Carbon Cycle Puzzle. So the goal is to create a low-cost deployment chamber to measure greenhouse gas emissions, especially during the wetting and drying phases of intermittent streams. We've seen there's some ways to do this in reservoirs, not a lot in streams, and certainly not in intermittent systems. And the Lycor is an amazing tool, but it is made for soil and it's very expensive. So we were looking to cap the price at about $1,000. So this is just the schematic of our system. So like LiDAR sensor would automatically sense the water level surface, and then the linear actuator would send a chamber down to the water surface or down to the collar if the stream is dry, and then measure methane and CO2 flux. And so this is a collaboration between myself, my advisor, myself. We're ecologists, biogeochemists, a biophysicist, and then two brilliant environmental engineers. So again, not to leave you hanging, but I will say my plug is come see us at AGU. I'll be presenting a poster on this work in the advances in measurement, synthesis, analysis, and modeling of greenhouse gas fluxes in natural systems, which is a poster on Tuesday. And with that, I will take any questions.
Thank you so much, Olivia. That was really exciting to hear about. As someone who has gone into the intersection of ag and intermittency, it was really cool to hear about your work. I'm not seeing any questions pop up in the chat. I will ask one then. You do have a compliment from Anne saying amazing study and ambitious study that was well presented. So, but I'm curious with your declines in concentrations moving downstream. Um, do you think that in the urban section where there's not much happening, is it is there something beyond hydrology that could be controlling this? Is it just nitrogen limited perhaps by the time you get to that urban area because so much has been processed out? Because those concentrations I think were relatively low.
Yeah, I will say we do see like the highest concentrations that we've ever measured were in Flat Branch, that like urban tributary where I'm doing my greenhouse gas work. But it's definitely possible. I will say what is kind of unique is that the urban, even down when we get to the urban section, there is still like a substantial amount of riparian buffer, so it's not as concrete lined as a lot of other urban systems.
Hmm.
But I think a lot of it may be explained by kind of just constant inputs. So I went through and mapped at one point just on Flat Branch every single pipe input, and it was like every 500 feet or less. So I think
Wow.
there is
Okay,
also
that's
just
a
a
lot.
lot of inputs. Yeah.
Cool. Super interesting. Well, I think that about wraps it up for today. And we're gonna have to end now since it is at time, but I just want to remind everybody that we have our workshop coming up in January, and our final seminar will be November 3rd, focused on the lower basin, featuring speakers from Arkansas, Louisiana, and Mississippi. So we hope to see you all there. And a big thanks to our speakers. I'll give you a round of applause, but I'm assuming everybody else on the call is also giving you one. So thank you, everybody, and we'll see you later.